Okay. Yes.
Are you going to post lecture 9 or just continuing for lecture 8?
So yeah, so I can post lecture 9, but it's really just going to be the end of lecture 8. So you guys have everything you need for the exam. Okay. This is on the exam. It's just the end of lecture 8 that I'm covering now, because if you notice I didn't finish lecture 8. So yeah, so don't feel like, oh my god, I can check out, this isn't on the exam. No, this is totally on the exam. Yes. Okay. For those who weren't paying attention, on Tuesday, open recitation this week, which means you can come if you want, you cannot come if you don't want, you can come if your Tuesday recitation, you can come if your Thursday recitation, you can come at 1, you can come at 2, you can come at 3, you can come at 4, you can, it's like, you know, we're all like getting wild and crazy. A lot of people did come and they spent a lot of time drawing and answering questions, and I think people thought it was helpful, right? Yeah, you were there, yeah. So we'll see. Exam 1 next week. Hopefully everyone started studying. I was walking, so here I'm going to be obnoxious. Can I be obnoxious? Yes. Okay. So I was walking to class or to work or somewhere the other day, and these 2 students were walking behind me talking. And the conversation went something like this. Hey man, how you doing? Like, it was one of those things, which okay, I understand, we're all like totally tired in the morning. I get it. But like, it was like a lot of expletives, which I understand too, because they don't have children and haven't learned to filter their mouths yet, but they were talking about how they're not studying for anything. And in a cool way, like, you know how in high school it was kind of cool to not study? Like, it was cool to be dumb and not study. But in college, you start to realize that's like, maybe it's still cool, but like, it kind of bite you in the butt later if you truly are dumb and not studying. And what I mean by dumb is you are dumb because you're not studying. So anyway, I kind of was like biting my tongue the whole way because I had to walk for a while with them behind me. So I heard their whole conversation, and it was all about how like, oh, the kids are in class and can't step. Like, oh my god, I wish I could just skip, but I gotta go. I'm like, why are you in college? Maybe they were freshmen, and maybe they were, but I'm just like, I am like so thankful. Well, so this is what I went in my head. I'm so thankful I have mostly juniors and seniors because you guys are so mature compared to that. I mean, yes, there's always someone out there who's like that, but like, it's not cool to be dumb. There's nothing cool about that. Anywho, so if you see people acting that way, please just go to them and be like, in 2 years, you will really be embarrassed that you're acting this way. Okay. Sorry, that was me being obnoxious and like also being a parent. And I'm like, please, like, I don't think I ever was like that, but I don't know. I was a slacker. I was, but I think I tried to hide it. Okay. Exam questions are due. That's I have a lot of public service announcements but I'm just going to space them out through the whole semester. Exam questions are due by 5 p.m. on Friday. So those of you who want to contribute, please email them to me. If it's even 501, I will ignore it. Remember, multiple choice has to, all answers have to be real answers, not fake answers. No quiz this weekend. I should give you a quiz this weekend because we're doing new material next week, and now you're not going to be prepared for lecture next week. But then I also thought to myself, you guys are going to be trying real hard to study, so maybe fun. Oral exam option. I got one email from someone who wants to do the oral exam. I saw that. I didn't respond, but I saw it. I'll respond later. If I ever don't respond, just keep pestering me. But please give me 24 hours. So just let me know. We do have to schedule that. It's not during the normal exam time because I need to be giving the exam. So we'll work that out. Okay, take 5 minutes to write. So this is supposed to be like a question that's going to kind of encompass the whole entire unit that we've talked about. So for those who have been studying, this is a good option, a good opportunity to test yourself and to see whether you can think big picture or not. Okay, so you're studying an unknown protein in a lab, in your lab. One way to develop a hypothesis about the function of an unknown protein is to determine its cellular localization. What I mean by cellular localization is where is it in the cell? Is it in the nucleus? Is it in the cytoplasm? Is it in the mitochondria? This can be estimated through analysis of the protein's sequence. So you can look at the sequence and hypothesize where it might be. So what would you look for in a protein sequence? Oh my God, I'm forgetting apostrophes everywhere. What would you look for in a protein sequence that would help you to predict the protein's localization in the cell and then therefore possible functions of that protein? So go to it. Okay. Oh, wait. Continue for a second. Okay, how's everyone doing? Okay, let's take the temperature of the class. Who felt like they knew right where to go with that? Good. That's good. Okay. So Sean McGovern. Oh, there he is. Okay. So tell me how you approach this. Or what you would look for. Pretty much, that's the first stop, right? Right. So first thing, look at the sequence, and remember, we looked at a lot of different signal sequences, or different biochemical characteristics within a protein sequence, right? And so just by looking for that, you can find out, is this going to be targeted to the nucleus? Is this going to be targeted to the mitochondria? Is this going to be targeted to the lysosome? All of those things. Very good. What other thing? Yes?
You can look to see connected to the amino acids and the chain to see if it's hydrophobic.
Yes, you could do a hydropathy plot and look for a transmembrane domain. Exactly. Yes? So you can look for where the signal sequence is, because some signal sequences are internal and are a transmembrane domain, right? Great. Yep.
You look for the presence of like NLS to see if there's
Yep. So you could call that a signal sequence in a way. It's not, but it has the same function because it's sending protein somewhere. So you can look for an NLS. Exactly. So that's really, these are all exactly what I was looking for. And so I can give you any sort of protein and some characteristics about it, and you should be able to tell me quite a bit. Yeah.
For the ones, the proteins that have the transmembrane domain, they're going into the ER, right?
Yep.
And are they getting stuck there?
Proteins with the transmembrane domain. That is a great question. They're going into the ER, into the ER membrane. Are they getting stuck there? Some of them are. Well, what kind of sequence, the ones that are stuck there in the ER, what kind of sequence will they have? KDEL. Right? So if you have KDEL, you're going to stay in the ER, either in the ER or in the ER membrane. If you don't have KDEL, then you're going to continue through the secretory pathway. Because don't forget, the membrane that is the ER membrane becomes the membrane that is the cell surface membrane. It is the same membrane. Yes?
So you could have a hydrophobic transmembrane sequence at the surface of the cell.
You can have a hydrophobic transmembrane sequence at the surface of the cell. Yes, that's what receptors are. Remember, receptors on the cell surface. People always draw receptors like, let's see. People always draw receptors. Here's our lipid bilayer, right? People always draw receptors like this, right? But that's not right. It actually has some sort of transmembrane domain, right? The part that goes through the trans through the membrane, and that is hydrophobic. So this might be your cell surface, plasma membrane. But that all starts at the ER, where it's made like that. Let's try that. Right? And then it buds, and it's still in there, and it goes to the Golgi. Oops. And it's still in there, and then it goes to the cell surface, and that's how it gets there. So I had a conversation with a student the other day who totally didn't connect that. So I'm so glad that you kind of brought that up, and that I got to make sure everyone understands that. Okay. Yep. Ah, we'll learn all about that today. Thank you for the introduction. Okay, so that's great. Does anyone want to add anything to that? Yes.
I have two, I don't know if you can use it. Can you look for the signal patch to bind a mannose-6-phosphate, and then look for the cytoplasmic tail sequence to bind a mononucleation?
Okay, can you look for a signal patch to see if it's tagged with mannose-6-phosphate? Probably. Right, so there's no actual mannose-6-phosphate yet, but in the protein sequence, so that's a harder one because signal patch originates from the folded, it basically is in the folded protein. It's in the sequence, but it's very hard to go from sequence to folded structure just by your eyes with a computer. There might be programs though. I believe there are programs that you can use to put a protein sequence in, and it will use an algorithm to search for something that would target it to the lysosome. Yeah. And then the other one you had was
For receptor-mediated endocytosis.
Oh right. So can you look to see if it's going to be endocytosed? Well, so that's harder because ubiquitin is added to lysines, and lysines are in everything. But I guess you could look in the cytoplasmic tail if you know what side the cytoplasmic tail is on, and see if there are lysines. Okay. Yep. About what? Same thing. What's going on here? Oh, there we go. Okay. Let's go on. So that's all very good. And so if you were able to kind of put that together, that means you're well on your way to kind of being organized and ready for the exam, because you're able to take all the details and put them together into something that's useful. Great. Okay, so the so far slide has increased in size again. The other day we finished up with receptor-mediated endocytosis, and we spoke about secretion, regulated versus constitutive. And then today we started nuclear import, and today we're going to finish nuclear import, export, and talk about mitochondrial import. Okay, so we were talking about how nuclear proteins, if they're supposed to go, remember they're translated in the cytosol, but they need to get to the nucleus. And how they have a nuclear localization signal, which is another one of those things that you could look for. And that nuclear localization signal binds to a nuclear import receptor. And so that's what this is showing here. The little S's, those are nuclear import receptors, and you can see that there's a binding site in the crook, I guess, for the nuclear localization signal. This is not how real proteins look, but you know. So the whole purpose is to facilitate import of that protein into the nucleus. And those nuclear import receptors, they bind to the NLS and the cargo protein, as well as these FG repeats that are along the fibrils on the nucleoporins. And so here you can see a protein with an NLS is binding to the nuclear import receptor, or importin, and then it interacts with those FG repeats and goes through the nuclear pore. And that requires RAN. And we're going to talk about exactly how that requires RAN. And then when you get into well, really what RAN does is it regulates the release of the protein inside the nucleus. And RAN is another GTP binding protein. So you guys should already start thinking to yourself, RAN, GTP binding protein. What other GTP binding protein did we talk about? RAB. How are they similar in their function? Right? You want to start comparing and contrasting those things. So RAN-GTP is going to bind the nuclear import receptor in the nucleus, and it actually changes the conformation of the nuclear import receptor, and it prevents it from binding tightly to the cargo protein, so it's released. And then it remains bound to nuclear import receptor and comes out of the nucleus. Right? Because you want that nuclear import receptor not to stay in the nucleus, but to go back out so it can shuttle more nuclear proteins into the nucleus. So you can think of your nuclear import receptor, your important, or your exportant as kind of like as shuttles for protein. So this picture is just showing exactly that. So you've got a protein with an NLS, and it's binding to your important. It's going into your nucleus, and RAN GTP. So remember, RAN is a GTP binding protein, so it's either GTP bound or GDP bound, and all that really is doing is changing the conformation of RAN and affecting its binding to other things. And so RAN GTP likes to bind to important, and that will release the cargo protein in the nucleus, which was the whole point. So then RAN can come out along with the important. Another GTP binding protein, just keep that in mind. And keep that in mind all the way through the whole semester, because there's a lot of them that we'll be seeing. Okay, any questions? Okay. So there's directionality. Oh, yep.
Sorry, so it doesn't actually lose the section of phosphate until it gets back into the cytoplasm, and then it releases the importance by changing its conformation.
Right. Yeah, exactly. So, and that's all dependent on RAN GAP and RAN GAF, and we're going to talk about that. Exactly. So thank you for the introduction of the next slide, because so RAN GAP lives in the cytoplasm. That's where it is. It never goes into the nucleus. Well, maybe it does, but from what I know, it's a cytoplasmic protein, and its job is to hydrolyze that GTP, the GDP and RAN, in the cytosol. And that's going to affect binding to importance, exportants, cargo, all of that. And so we'll go through the whole thing, and you'll see how it's going to affect that. And then there's RAN GEF, and that's the one that kicks off GDP and adds a GTP. That actually is localized to the nucleus. It lives in the nucleus. It actually interacts with the chromatin. And so this provides some directionality to the nuclear import. So things are going in the right direction. Important's going in, dropping off cargo. Exportant's going out, dropping off cargo. So nuclear export requires a nuclear export signal, and a nuclear export receptor. Very similar to nuclear import. There's a different sequence and a different receptor that's going to be shuttling things, and it also requires RAN. So with nuclear export, you've got your exportant that goes into the nucleus, and it's going to recognize a protein with a nuclear export signal. And in this case, RAN GTP is actually encouraging binding of the exportant with your protein that needs to be exported. And then it brings it out, and then RAN GAP is there, and it hydrolyzes GTP to GDP, so conformational change, which releases the exported protein in the cytoplasm. So I have a lot of figures showing this because I think the first time I learned nuclear import and export, it seemed really complicated. So what I encourage you to do is look in our textbook and read what's there. And then go on NCBI books and put in your search terms nuclear import or nuclear export and read whatever they give you. So this comes out of a different textbook because I thought their figures were really nice. It's called the Cell Molecular Approach, and it's freely available on NCBI books. And sometimes I like to go to that textbook to read about whatever topic because I like the way they write for certain things. Okay, so RAN is a big part of nuclear import and export. It's regulating the binding and or release of cargo from the import or export receptor. If you have no RAN, then the cargo won't be released won't be brought to where it needs to go or released where it should be. I want to warn you, when you're studying, don't confuse RAN GDP and RAN GTP with RAN gap and RAN gap. People do that all the time on my exams, and they get it wrong because they're telling me RAN gap is binding to importance and stuff like that. So make sure you really understand what each one is doing so you don't confuse those, because they're very easy to confuse because they all are RAN something, right? So this is showing, on the left it's showing import, and on the right it's showing export. And so here you have your protein with your NLS, and it's binding to your nuclear import receptor, and it's going in, remember, hopping on those FG repeats, going into the nucleus, and when it gets in there, you have RAN GTP hanging out. Why? Because there's a RAN gap in there. So it's always keeping all the RAN GTP bound, because that's its job. And so when that binds, it's going to bind tightly to the nuclear import receptor and take away the binding site from the cargo, so the cargo is released. So if you can kind of keep in mind that it really has to do with the conformational change, right? So then you have your nuclear import receptor bound to RAN GTP, and it goes out of the nucleus because it has its own nuclear export receptor. And then when it comes out here, what do you have? You have RAN gap. What does RAN gap do? It hydrolyzes GTP on RAN. And so it's hydrolyzing the GTP. Again, conformational change, and you have a release of something. In this case, your nuclear import receptor. That's important, right? Because you want your nuclear import receptor to be sent back out so it can bring more things in. If it just hangs out in here, in the nucleus, that's not going to be very useful for imported proteins, right? So for nuclear export, it's kind of similar, but just reversed, where you'll have a protein with a nuclear export signal, and then you have a nuclear export receptor, and that RAN GTP is promoting binding. And so then they leave the nucleus, and then when you get out there, you have your RAN gap, which is going to hydrolyze your GTP to your GDP, conformational change, releasing everything. And then your nuclear export receptor can go back in, and it has its own nuclear import receptor that it associates with to bring it back in. Questions about this? Yes?
So is the only difference between import and export with RAN GTP is in import, it's only binding to one partner releasing the cargo. Then in export, it's binding to everything. I don't know.
I'm not following.
I'm not.
Yeah, so I think the important thing is sometimes if you try to compare the two, it can get confusing. It's important to compare the two, but in this specific situation, try to walk yourself through it and keep in mind that the goal is getting your NLS-containing protein into the nucleus and released. And so the binding between your nuclear import receptor and your NLS, that happens just in its normal conformation. And what RAN GTP is doing is it's binding and changing the conformation. I'm going to show you a slide in a minute, and that releases it. And with export, it is different because in this case, RAN is kind of encouraging the binding. So I think that's what you were seeing a little bit, that kind of difference. So RAN GTP is actually encouraging the binding between the nuclear export signal containing protein and the exportin. And then it goes out, and their hydrolysis is releasing it. Yes?
Does that imply that both are occurring at the same time?
Everything's always occurring because you have to remember there's not just one protein in the cell that needs to go one place. Tons of proteins are constantly moving everywhere in the cell. So really, this is happening times 1,000 at the same time.
I mean, what's happening, what I was trying to figure out is that would the import occur, and then once you have that RAN GTP, something would be going out automatically? Like, or would it happen?
So are you wondering whether that RAN GTP here
Yes.
is going to then be used here?
Yes.
I would make that assumption because you know what, all RAN is RAN GTP in the nucleus, and that's because RAN gap lives there. So the nucleus is stocked full of tons and tons of molecules of RAN-GTP that can always be used for either, and it just depends on what's present. Yeah.
So brand GTP is the inside of the nucleus. It goes out, gets one of the phosphates cleaved off, becomes GTP. Is this cycle does it recycle like?
RAN's going to go back in. So RAN-GDP is then going, it has its own nuclear import receptor and it goes back in. Yeah. It needs to always be brought back in because the gas is in the nucleus.
Okay.
And that's the way it gets recharged, I guess.
Okay.
Okay. Who else is raising their hand? Yes.
So are RAN-GTP and receptors both essentially recycled?
Nuclear import receptors, nuclear export receptors are all recycled. They kind of shuttle things in and out,
And then
along with RAN.
Okay.
Yeah, RAN is constantly being recycled and constantly going back and forth between GDP and GTP. And what state it's in is depending upon where it is because its gap is on the outside, its gap is on the inside. Yep. Any other questions? Can you imagine being the person who worked this out? Like scientists actually worked this all out. It's pretty wild when you think about it. Pretty intense mechanism. Okay. So remember I was saying conformational change, right? So when your nuclear import receptor comes in, brings its cargo into the nucleus, it needs to release it, right? And so what happens is when RAN-GTP binds, it changes the conformation. So you could see right here, here's this blue part is your nuclear import receptor, and the green is RAN-GTP, and then there's this loop right here that's part of the nuclear import receptor, and that changes depending on whether or not RAN-GTP is there. So if you have your nuclear import receptor, so you've got this loop binding to your NLS. And when it comes into the nucleus, RAN-GTP is going to bind, and it's going to take the place. And you see this loop is going to switch and bind here. And that's what releases the cargo protein. And so when I talk about conformational change, it really has to deal with the part of the import receptor that's binding to the cargo protein doesn't exist anymore once RAN-GTP binds. Well, it exists, it's just in a different conformation. It's not available for binding anymore. And that's how it's released. Questions? Okay. So let's apply this to something. Remember I was saying at some point that transcription factors go into the nucleus to bind to promoters and activate gene expression. And gene expression essentially is regulated by whether or not that transcription factor can get into the nucleus. And so it's all about keeping the transcription factor out of the nucleus until it's ready to go in and bind to the promoter in many ways. And so I showed you that movie where that transcription factor called NFAT, NFAT, I don't know if I called that. Did I call it that? Where you add calcium and it all went into the nucleus. Do you guys remember that from Tuesday? Green into the nucleus. The video is on there, so you guys can watch it at home. So this is kind of telling you what's going on here. So basically, the nuclear localization signal is wait, let me make sure I get this right real quick. I always get turned around here. So nuclear localization signal, okay. So nuclear localization signal is, they're calling it a nuclear import signal, is normally phosphorylated. And so when you have high calcium, remember they increased the calcium in that video, then a phosphatase comes along and removes those phosphate groups, and that exposes the nuclear localization signal. And then it can interact with the nuclear import receptor and go into the nucleus and activate gene expression. With low calcium in the nucleus, then that transcription factor can be phosphorylated again because you'll have a kinase that becomes activated, and then you cover up the nuclear import receptor and you expose the nuclear export receptor, and it can go up, so then it can be exported out of the nucleus to turn off gene expression. So that's that same thing that was on the video. And the take-home message is, this is a common mechanism. You guys might think, why do I care about this one signaling pathway? It's an example. It's one mechanism, but a very common mechanism for how nuclear imported proteins is regulated. And it needs to be regulated when you're talking about activation of gene expression, right? If you don't regulate it, then how is gene expression controlled? Then you should just express all our genes all the time. It seems kind of silly. So also NF-kappa B uses the same mechanism. And so if I ask you, what's a mechanism for proteins to do blah, 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 a very common one that you can always use is, oh, something hides the nuclear import signal, nuclear localization signal, and activation of some pathway is going to expose that nuclear localization signal and allow that protein to go in. Which I can't tell you how many times that mechanism comes up in biology. So it leads me to the practice problem that you guys are going to do right now. So you're studying a viral protein. I always like viral proteins because they mess with things. This viral protein appears to be facilitating the import of certain proteins into the nucleus. So that means that certain proteins, only when this viral protein is present, are imported into the nucleus. So what do you think is going on? So first form a hypothesis, and then design an experiment to test your hypothesis. And so the other side of that is there's another viral protein that appears to be preventing nuclear export of a protein. What could be going on here? How would you test your hypothesis? So form a hypothesis and then test it. So take a few minutes to talk to your friends about how you would approach this, and I'll circulate and help whoever needs help, and then we'll discuss. some interesting feedback. So some people were like, well, I don't know how to approach this. It's okay. So maybe I wasn't giving enough. So I'm going to rephrase the first part. Just think. Just use your brains for a minute. Think, listen. Sometimes writing throws everything off. So viral proteins induce gene expression all the time. And so if you remember from genetics or molecular biology, that usually means some transcription factor is being sent into the nucleus. That shouldn't be, right? Because the virus is forcing it. And so I'm saying some viral protein appears to be facilitating import of a protein, some protein, into the nucleus. So we'll call it a transcription factor. It's forcing a transcription factor into the nucleus to activate gene expression for its own purposes. And so how might that viral protein be behaving? What might it be doing? And how would you test that? Yep.
For the first part, you could find like an NES on the viral protein.
An NES or an MLS? Exporter.
Oh, NLS, right.
Okay.
And if there's like buildup, you can use like radioactive tag. If there's buildup in the nucleus,
Okay, so your first step would be, well, let's see if this viral protein even goes in the nucleus. Look to see if it has an NLS, and then see if it goes there. And how would you test to see if it was actually an NLS? See, it goes there, but you know it's because it has an NLS. What would you have to do to that protein? How would you manipulate that? Great, you can remove the NLS and see if it stops going into the nucleus. Exactly. So that's one. That would kind of be part one of how to evaluate what's going on. So what else could we do to really find out how this viral protein is making the transcription factor go to the nucleus? Yeah.
Can you just remove it and like label the other proteins and see if they still making it?
Okay, so this is you already know this protein is making the protein get into the nucleus. So you're saying remove it and see if it doesn't go in. Well, yes, so that would be confirming. That would be confirming. So you always have to ask yourself, the experiment I designed, what question is it answering? And so that's answering the question of, does this viral protein force this other protein into the nucleus? And so that would be exactly how you would test for that, is you would delete that viral protein and see what happens to your nucleoprotein. Yes?
Probably modify the viral protein to see if it is an NLS or work NLS that forces items.
Okay, so modify the viral protein in some way to see if it is an NLS. That's kind of similar to what you're saying there, is delete it. But so we've made a horrible mistake, though, in how we're approaching this. We're designing experiments before developing a hypothesis. So that's, whenever you're doing research, you need to develop a hypothesis and then design your experiments to make sure you're testing your hypothesis. So what is our hypothesis? Yep.
It's probably doing something to a signal pathway in the previous slide, so it's uncovering a nuclear localization.
So it might be uncovering a nuclear localization signal. So it might be affecting the signaling pathway, resulting in uncovering of the nuclear localization signal. So that's one. What else might it be doing?
I think the virus is probably binding to the protein that's being imported, then the virus binds to it and then like whatever's blocking it so that it has no choice. Well, it's being told to go in.
Okay, so maybe the viral protein is binding to this protein and kicking off its inhibitor in some way, so exposing the NLS. So that's another thing. So it's kind of similar to what Eric was saying. What else? Did I see you raising your hand before?
It could be affecting RAN, GDP, phosphorylating.
Maybe it's affecting RAN, GDP, phosphorylating. Well, the problem there is that should affect all of nuclear import. So if you saw all proteins, not all, but like you saw an effect of global effect, then you could start looking at RAN. But you're looking at this one protein. So, does another hand go up? Yeah.
Does is it the same translocon on ER as well as the SAR?
Oh, so I just got a question was, is the translocon on the ER the same as the translocon on the nucleus? No. The ER has a translocon, the nucleus has a nuclear pore. And so the ER, you have a protein brought through SRP and SRP receptor, and then the proteins get through the nuclear pore through interaction with the importance that interact with the FG repeats on the nucleus. Okay, so let's see. So what I was thinking, so these are all really great hypotheses, and you could develop ways to test it. One is maybe this viral protein isn't important that binds specifically to this transcription factor, for example, or some protein. That would be a very obvious way to do it, right? Because then, boop, you're in. Or maybe this viral protein is an adapter protein that binds to the transcription factor and recruits an important. Again, same thing, because then the viral protein can have an NLS. So you don't have to worry about uncovering an NLS. And then maybe the viral protein is affecting the pathway and that is resulting in dysregulation of that mechanism for keeping the transcription factor out of the nucleus. So this is what you do. You sit and you think and you come up with all these hypotheses, and they are all testable, and then you design experiments, which usually result in modifying your protein and seeing if what you think is happening goes away, right? So good. Okay, so the second part was another viral protein appears to be preventing nuclear export of a protein. What could be going on here? How would you test your hypothesis? Yes? So calcium, it's keeping calcium levels low and hiding the binding site. So be careful with, okay, so this last, so what you're doing is right in the sense that you're trying to apply what you've learned before. And that's how you do research, right? So you're saying, if maybe calcium levels are low and it's hiding the binding site, we'll be hiding the nuclear import signal. So that's a very specific pathway. And so you want to be careful with getting too specific with your pathway that you're relating it to because it may or may not apply. But maybe you said the binding site is being hidden, right? So what binding site? That's related to nuclear export. What binding site is required for export of proteins? What binding site is required for export of nuclear proteins? What sequence? Nuclear export signal, right? So maybe it's kind of related to what you were getting at. You were getting very, very specific, but maybe something is causing your nuclear export signal to be covered up. Maybe your viral protein binds to the nuclear export signal. That's a hypothesis. Maybe calcium is playing a role. Maybe you can start to evaluate that. Did anyone have any other thoughts? So science is all about just guessing. If it's a logical guess that makes sense, then you just test whether it's happening. Right? Okay, that was fun. Import into the mitochondria. So remember, and I have the chloroplast up here. We're not really paying attention to that because I just don't cover it. But remember, your mitochondria has two membranes, right? You've got your outer membrane, you've got your inner membrane, and then you've got an intermembrane space, and you've got a matrix. So there's a lot of places for proteins to go, right? So you have to think to yourself, oh, they must be regulated in some way, right? Because it's not as simple as the ER where it's like in, you're in. Here, what if you're a protein that is a membrane protein that's an outer membrane? What if you are a protein that works in the intermembrane space? What if you're a protein that works in the matrix, or you're an intermembrane protein, right? So different proteins are going to reside in different compartments, and they need to get to the right place. So the key players here, signal sequence, translocators, chaperones, and energy, namely ATP hydrolysis and proton gradient. And so, does this sound familiar? What other organelle uses a similar mechanism for protein import? ER. There's so many things that are almost exactly the same. So that should make your life easier. There's just that extra membrane to deal with, and that extra intermembrane space. So when you're studying, it might be useful to compare and contrast import into the mitochondria and ER, so you can think about them and look at what's similar and what's different and make sure you're not confusing them. So proteins, they're post-translationally derived into the mitochondria, so that's different in one way. With ER, you can have both post-translational and co-translational import. But here, with the mitochondria, only post-translational import. So you're going to have translation in the cytosol, and then there's going to be this mitochondrial targeting sequence, which is not a specific sequence, but it's a specific structure, and it's with specific biochemical properties. So the amino acid sequence, remember, amino acid sequence dictates structure. So the amino acid sequence was going to form a helix with positively charged amino acids on one side and hydrophobic amino acids on the other side. So it's going to look like this, and it's going to be recognized by a specific receptor. And just like import into the ER, this signal sequence, we can call it a signal sequence, is cleaved by signal peptidase inside the mitochondria. So this is what the signal sequence of alcohol dehydrogenase looks like. So alcohol dehydrogenase is a mitochondrial protein, and so it needs to be targeted to the mitochondria. It needs to interact with that mitochondrial import receptor. And so here, this big blob, the gray blob, is your mitochondrial import receptor. And this helix, you see how it's kind of wound around with side chains sticking off? It takes practice to look at protein structures, right? Right? Let's practice just for a minute. Right? So this is just, your space-filling model of your mitochondrial import receptor. We don't really care so much about anything other than the fact that it's got this binding site. See that groove right there? And that helix fits right in there. And these amino acids that are sticking off that side of the helix, because remember one side needed to have one property, the other side needed another biochemical property. So that side right there has a biochemical property that mediates interaction with the binding site of the mitochondrial import receptor. So you can guess that if you're trying to study mitochondrial import and you think this might be a mitochondrial targeting sequence, an obvious thing to do would be to look for a mitochondrial targeting sequence and mutate it to see if it won't, so if it doesn't bind anymore, it's going to get imported into the mitochondria, right? Does everyone agree with me? You have to agree with the professor, right? There's really no way around it. Otherwise you fail. No, just kidding. Okay, I'm just kidding. I'm not going to fail anyone. I don't believe in failure. These are all winners. I don't know. I don't know. So you have translocons, just like import into the ER. And so here you have multiple translocons. And that's how the protein gets in. And there's Tim and Tom. That's how I always remember mitochondrial import. And you can remember what's what because Tom is translocase of the outer. That's what the O is for. Outer membrane. So that's Tom. There's also Sam. Sam hanging out with Tom. And then there's translocase of the inner membrane. So that's Tim. And so you shouldn't confuse those unless you're straight memorizing without doing any thinking. That's why I warn you, please don't memorize without thinking. So you should always know Tim and Tom. And so Tim is the translocase of the inner membrane and is also hanging out with this OXA complex.
What is Sam's name?
Oh, I don't know. Sam's. Something. It probably does. It's just another. So these are all translocons. There's multiple. So So with the ER, we really just talked about one kind of translocon, and I said, don't worry, translocon getting inside. Here it's kind of the same, you just need to understand that you have to get through two membranes. So there's going to be two kind of classes of translocons. So, yep.
So when you talk about like kind of transmembrane protein and here you have one in with a transmembrane protein and one in the membrane.
Yeah, so you can have a transmembrane protein with KDL, so with an ER transmembrane protein. Yes, you can have a membrane protein in the mitochondria. Well, think about, okay, so here's where I'm pretty bad. But think about what the mitochondria does. What's a major function of the mitochondria? Producing ATP, electron transport, oxidative phosphorylation, all those things, right? That I don't teach you in this class, but you learned before. So, do those things just magically happen, or do proteins participate in those processes? Right? So every process that you've ever learned that you were just told, hey, memorize these structures, this becomes this, becomes this, becomes this. Well, if they don't just doesn't magically happen, proteins make them happen. And so all these proteins are living in the mitochondria, and some of them are membrane proteins that are in the membrane, right? Can anyone think of a membrane protein that is in the mitochondrial membrane? Isn't there there are protons being pumped in? Yeah, protons being, so there's membrane proteins that are pumping protons in, right? And making that proton gradient. Yeah. Think back, you learned about it, you saw pluses and stuff. Proton pumps, yes, exactly. In the membrane of the mitochondria, it's got to get there somehow. This is it. Okay. So, major steps of import into the mitochondria. It's going to look, this is your simple way, right? It's going to look very similar to what you saw with the ER, right? Protein with a signal sequence binds to the receptor that's part of this TOM complex, and it's going to go in, then it's going to interact with your TIM. So in this example, this is a protein that needs to go to the matrix. And so it's going to bind to TOM, it's going to start going through, it's going to get, and then it's going to interact with TIM and be brought in, and then you're going to get signal peptidase that's going to cleave that signal sequence. And then you have your mitochondrial protein in the matrix. So that pretty much almost looks exactly like ER, right? Except for you're going through two membranes. So ATP hydrolysis and a pH gradient drive transport across the mitochondrial membrane into the matrix, right? So you're going to have chaperones that are binding to your mitochondrial protein and keeping it loosely unfolded so it can make it through. Because a big globular folded protein is going to have a hard time making it through the translocon. So binding and release by HSP70 requires ATP hydrolysis. So that's one place that ATP is used. And so then you also have this proton gradient that's in your intermembrane space, and that's helping to drive the protein through the TIM complex. And then you also have mitochondrial HSP70, lots of chaperones. Remember, there were lots of chaperones with the ER too. Mitochondrial HSP70 is helping to kind of pull the protein all the way in. So there are two models for how mitochondrial HSP70 drives protein import. Both require ATP. One is called the thermal ratchet, where you just have, basically what's happening is thermal ratcheting. You can imagine just from this thermal motion, the proteins are kind of moving inside they're in the translocon, it's kind of moving. And every time it moves down a little, an HSP70 is going to bind and kind of preventing it from moving back up. And so that kind of hand over hand motion of the HSP70 binding is what brings it in to the matrix. The other model is this cross-bridge ratchet model, where the actual conformational change that occurs from ATP hydrolysis of HSP70 is pulling the protein through. You know, I'm not sure which one is right, but that's two ways that HSP70 can be helping to bring the protein into the matrix. Okay. Okay, so then you also have, so there are different, so this is actually a very nice figure, so I encourage you to look at it in chapter 12. And it's showing the different types of proteins and how they get into where they need to go. And so it gives you an example of a membrane protein that's an inner membrane protein. And so it's a protein that actually lives in the inner membrane, in the mitochondrial inner membrane, and it has a stop transfer sequence, similar again to ER. And so it's got to be imported through TOM and TEM, and then once it hits that stop transfer sequence, it moves over into the membrane, and that's where it lives. Also, there are proteins that need to end up in the matrix space. There's also proteins that are in the intermembrane space, and so they'll also have a stop transfer sequence, but then they cleave from it, so they're soluble in the intermembrane space. And then TIM22 complex is involved in insertion of proteins with transmembrane passes into the inner membrane. And so again, it's quite similar to what we saw with the ER. There's going to be these hydrophobic transmembrane domains, and as they kind of come through, they're going to move over into the membrane. And then porins, those are inserted into the mitochondrial outer membrane. And here, what actually happens is you have import of your protein through your TOM complex, and then in your intermembrane space, chaperones are binding it, helping to keep it loosely folded, and then it's kind of fed back outwards through the SAM complex, and it is inserted into the membrane that way. Just a point of interest. The SAM complex is conserved among bacteria and eukaryotic cells, so that's evidence supporting the endosymbiotic origin of mitochondria. Happy studying. Come to recitation if you have questions.